Okay, thank you very much, Tracy, and thank you for the opportunity to talk about the book today. It was published by Houghton Mifflin Harcourt on May 11th. And I'm a big booster of local bookstores such as Green Apple. And we went in there this weekend and they actually had the book. So I was amazed by this actually. So I got friends of mine and colleagues to write some endorsements for the book, and they appear on the jacket. One of them is our former Chancellor, Sue Hellman, and the other is an old friend of mine from the NIH, Florence Hazeltine. So this was one of the parts that was really fun, connecting with old friends to see if they would support this effort. And I won't read it, but this is how the book is being marketed. The indispensable guide is being marketed to the general public. I thought I'd say a little bit about how this book came to be. In many ways, it was a very happy accident. The NIH had decided several years ago that the placenta was understudied. So they decided to sponsor a human placenta project, or the HPP. They invited me to speak at the opening and many subsequent meetings. And to the opening, they invited members of the general public and reporters, including a reporter from the New York Times, Deb Grady. And I happened to be in a discussion group where she was, and afterwards she talked to me about coming to visit our lab and seeing what we did with the placenta. So Deb came and she ended up writing a story for the New York Times on the placenta. And they had taken a lot of pictures and a lot of notes on what we did. And they featured us prominently in that article. So after it was published, I received an email from a book agent who said, you know, these are all ideas that I haven't really heard before. Would you think about writing a book? And this woman's name was Kitty Cowles. And I said, well, why don't you come to my office at UCSF and let's talk about this. So she came and we had several meetings. I was really reluctant to take on this big project because of my day job, which usually consumes all day, some nights and some weekends. I wasn't sure how I was going to fit this book in, but Kitty thought it would be a good idea just to see if it was going to fly. So she asked me to write a book proposal. So I wrote outlines, the first chapter and outlines for the chapter, all the chapters. And Kitty took it to publishers, and Rux Martin at Houghton Mifflin Harcourt actually bought the book, which I found at the time astounding, and I still find this astounding. Part of my astonishment is that Rux Martin is famous for having published many of cookbooks of very famous people like Mark Bittman. And why she When for this book, I'm not sure. This is well, I guess you could consider it cooking pregnancy, but I was really astounded that she would want to buy this book. Halfway through, she retired, and Deb Brody, who is now the publisher at HMH, took over, and she was incredibly helpful. So this is a little bit about how the book began and how it was finished. I thought I would just give a little summary of what's in all the chapters, and I hope that people will ask questions along the way. I don't want this to be a formal lecture, so if someone would watch the chat, that would be great, and ask questions as they occur to you. The first chapter is about the beginning 8 weeks of development, which embryologists call embryogenesis. And I think what most people don't know much about human development find astonishing is that essentially the baby is entirely formed by the first 8 weeks. This is somewhat of an oversimplification, but it's also mostly the truth. You can see over here, maybe I'll grab my laser pointer. You can see over here a drawing of a human embryo at 48 days. And this drawing is in the book. So this is by way of introducing that I decided pretty early on to put drawings and artwork in the book. Having taught anatomy for decades, I realized that a picture is worth 1,000 words. And I find this picture of an 8-week embryo to be fascinating because you obviously their head, their limbs with digits, and their eyes and ears, and all the internal organs have formed. And there's so many implications to this. The major implication is that most of the major birth defects occur within the first 8 weeks of pregnancy. And for a decent amount of this time, most women don't even know they're pregnant. So this means that you need to prepare for pregnancy. If there are things you're doing that you think may not be good for the baby, then the time to stop those things are before you get pregnant. So it's really, I think, important. Excuse me, my phone is going crazy. It's really important to realize the speed at which development occurs, because after the first 8 weeks, essentially that template for the baby grows. So it's all about growth. And a lot of women, I think the baby is so small and very protected that nothing can happen to it. And that's the farthest thing from the truth. I also talk about some things that we think can harm babies. Some of this, and this is true for a lot of the book, I think it may not be what people want to hear, but it's definitely scientifically valid. And one example of this is alcohol use. I think I have had this experience many times, and I'm sure that clinicians on this phone call have had this experience many times, that I've been at parties where there are pregnant women and people know I work on pregnancy, And the husband comes up to me and says, tell my wife that having a few glasses of wine isn't going to hurt. And I think these kind of experiences are what has really stimulated me to write about this and the fact that alcohol can hurt. It's one of those things like radiation that we don't know the dose for people and for developing babies that is safe. And so the safest thing is not to use alcohol during pregnancy. And I think one of the interesting things around this is that Italy has just issued guidelines about not drinking during pregnancy, which is, I think, quite amazing for most people who are used to thinking of Italy as one of the reasons why pregnant women can drink during pregnancy. The other reason is that there are astounding rates of fetal alcohol syndrome. And we know this now because fetal alcohol syndrome can manifest oftentimes very late in development, even the teenage years, as learning disabilities. It's really hard to diagnose for many, many years. And the rates are quite astounding if you consider problems that children have off into the teenage years. Smoking is another problem, and I think it's important to emphasize that smoking also diminishes semen quality in men. Cannabis use is really becoming a major issue. The American College of Obstetricians and Gynecologists estimates that about 3 to 5% of pregnant women use some sort of cannabis product during pregnancy. And we've just found the receptors for cannabinoids, or the active ingredients in cannabis projects, in placental cells. So this really makes you think that the placenta, just like the brain, can respond to marijuana, for example. Then there are also issues around prescriptions and over-the-counter drugs, which ones can get across the placenta to the baby, and the impact of infections. We've been in the last few years exposed to Zika, COVID-19 effects. And finally, the importance of vaccinations. In this country, only about half the pregnant women who are pregnant get a flu vaccine during pregnancy. And I wrote this long before COVID came along, and the vaccine hesitancy of women to get COVID vaccines during pregnancy, which could have been predicted from the reaction to getting flu vaccines. So there's a lot of work that needs to be done in this area. The second,
hey, Susan.
Yeah.
Yeah, could you talk about e-cigarettes? There's a question about that, especially since we're seeing a big, like, we've been reasonably successful on laws and policies that have reduced smoking, but there's been a sharp increased rate in e-cig use, particularly among younger people, and what that means if there's.
Yes, I do talk about that in the book, and the e-cigarettes have effects above and beyond tobacco use, as we know, because we know people have even died from using e-cigarettes, which now is attributed to mixing with oils such as vitamin E. So the e-cigarettes are incredibly dangerous. And there's some estimates now that if you look at past month smoking by pregnant women in the US, in some groups, the CDC estimates it's as high as 15 to 17%. And probably, I mean, I haven't seen the statistics on this, but it could be driven by e-cigarettes, which have, you know, the appearance of being less harmful because they're less noxious. So the next chapter is on the placenta, which I am just obsessed with. So I'll try to keep this short. The placenta is what we've studied forever. And most of the chapters have a vignette in it, a story. So it's just not facts and facts and advice and advice. And in this story, I tell how I became interested, in this chapter, I tell how I became interested in studying the placenta. And I won't go into that here, but I was hooked from the beginning. The first time I heard about what the placenta could do, I was like, I'm going to study that. So obviously I did. This chapter also covers development, and I think the placenta is just remarkable because its specialized cells called trophoblast cells are formed by day 6 after fertilization. Day 6, and these cells have already acquired what they need to do, and that is they mediate implantation. They're the ones that attach the pregnancy to the uterus. And we've actually worked out the mechanism by which we think this happens. But by 3 weeks, the placenta, even more quickly than the baby, has assumed its mature structure. And here is another drawing from the book. At week 1, our formation of the trophoblast cells, which are the plain gray areas that are on the surface of the chorionic villi. By week 2 after fertilization, the trophoblast cells are filled with undifferentiated mesodermal mesenchyme type cells, which are structures, give it structure. And I always liken this to the internal part of a tree. And then it acquires its blood vessels, its vascular feed in the 3rd week of pregnancy. So you already by week 3 of pregnancy have these little trees formed that will extract nutrients from maternal blood and return waste products to the same place. And the placenta is absolutely required for normal growth of the baby. I think it's quite interesting because there's so many things if we learn about the placenta in a pregnancy that it can really help a woman understand what's going on. And the first thing I think it's really important to know is the position of the placenta. The placenta is normally supposed to be attached up high in the uterus. And many times you see that the placenta actually attaches too low. And these conditions where the placenta attaches near or close or covers the cervix, which is the outlet of the uterus for birth, those conditions can be associated with a lot of problems in pregnancy. You obviously don't want the placenta to be born before the baby. So the placenta is the baby's lifeline. And if the baby loses the lifeline, then this is no good. So it's really important to know where the placenta is, which is easy to determine if you have good prenatal care. The other thing is if you think of the placenta as the seed, you also need good soil. It needs a good uterus to implant in with a healthy lining or endometrium. The other thing that I find really super interesting is the incredible ability of the uterus to expand as the placenta grows. The placenta is growing, the baby's growing, and this little tiny organ that people often compare to the size of a pear, this little tiny organ expands, has a capacity to expand to hold a baby, which is pretty remarkable when you think everything that's involved in that. The other thing is, and I'll talk about this later, is the placenta plays a huge role in regulating maternal metabolism. And we now attribute much of the cause of morning sickness to placental products. It used to be thought that a big driver of morning sickness is HCG, but that idea is waning a bit. It came about because HCG levels are highest in peak when most women are most affected by morning sickness. And we now know that certain products such as growth and differentiation factor 15 can account for morning sickness. And this work is quite interesting and has grown out of the fact that some of these molecules are being given therapeutically for other reasons, not during pregnancy, just to people in general, and morning sickness is recreated. The other thing that I think it's really important to understand is that most evolutionary biologists think that it's actually the placenta that made us human. In many other types of animals, placentation, which I've depicted here, is very, very shallow. The placenta lies on the surface of the uterus. And this is true for ruminants, for pigs, for dogs, and they also have different shapes of placentas than humans. But the most significant difference is that the human placenta, shown in dark gray here, invades extremely deeply into the uterus and actually lines a mother's blood vessels during pregnancy, which gets us the blood we need to grow babies. So you can imagine that removal of a very superficial placenta at birth is no big deal. But removal of this placenta that's deeply embedded in the uterus is a whole different story and a much more complicated process. And that's why birth in humans is such a much bigger deal than many animals. So the third chapter.
Hey, Susan?
Yeah.
Can I ask you a question about that placenta piece? Could you talk
Yeah.
about, well, let's do animals that are closer related evolutionarily to humans. Do they have, say like a chimpanzee, does it have a more like placenta to humans? That's just sort of an interesting, I don't know, just very interesting component of evolution.
It's a good question. And there's of course a huge amount of work done on our monkey placentas and in the great apes, are all of our closest relatives. And the answer is that the placenta is quite different in us. There's more invasion. We lose our stem cells in the placenta. Humans lose them. And for example, rhesus macaques keep their trophoblast stem cells. And we think that's why the placentation is so much more shallow in the, for example, the rhesus macaque, because if they need more invasion and more blood, they can launch more stem cells to accomplish that. But we cannot. What we get at the beginning of pregnancy is what we end up with. So we don't have any, we've lost our reserve. We burn it up in this deep placentation, which is perhaps not the best idea in the world, but it's what happens.
Thanks.
So the next chapter.
Ask another question, Susan?
Sure.
This is Diana. I'm wondering if using like complementation assays, is there, I mean, just backing up, because the mouse placenta is such a insufficient model for the human placenta, are there any assays existing, or, you know, can you think of them that would be like complementation assays in mouse using human trophoblast cells?
Yes, well, as I'm sure you know, Diana, it's very controversial about putting human stem cells into mice. We actually haven't thought very much about doing that because the human trophoblasts are so invasive. They're as invasive as a tumor. And it's likely that the mouse uterus doesn't have all the equipment that the human uterus has for fending off these tumor-like trophoblast cells. But it would be very interesting to see what happens. You can make an equivalent of teratomas from trophoblast cells, and they form extremely aggressive tumors in mice.
Hey, Susan, can I ask a totally ignorant question? This is Jennifer.
There you go.
Like, do twins have their own placenta? Is there a difference between like animals that have litters versus like humans that tend to have only one offspring?
Yeah. So it depends whether or not a human baby, when they're twins, has a placenta, its own placenta, depends when the embryo split. If there were two embryos, then obviously there are two placentas. But if the split occurs very early in pregnancy, then the embryos, two embryos or more, share a placenta. I mean, I think this capacity of the uterus to accommodate placentas was most dramatically demonstrated by the Octomom, right? She gave birth to 8 pretty good sized babies, each of which had their own placenta, which is just remarkable.
Susan, someone's asking, what does the deep embedding enable? What's the purpose of the deep embedding?
So the deep placentation enables a very big blood supply. So by term, there are hundreds of mils of blood per minute flowing by the placenta. And I mean, it obviously requires a huge amount of oxygen and food to feed babies, growing babies. And this deep placentation modifies a mother's blood vessels so that they are interrupted, and that interruption brings the blood to the placenta and then allows it to flow back again. It's a circuit that inserts the placenta into the mother's vascular system. So you get, if you're a pregnant woman, you have this new organ that's almost the size of the liver inserted into your bloodstream, which is absolutely, I still find this to be an absolutely astounding fact. So the consequences of this huge blood supply are really enormous, and many evolutionary biologists think that it enabled growth of our brains. And this is why in humans, the human cortex has doubled compared to lower animals. We have more of the neurons that we think with. And a lot of people think that this enormous blood supply that our deep placentas obtain for us is the reason why the brain had enough energy to double the cortex. Does that answer the question?
I think so. But
Okay, good. So I'll go on to the 3rd chapter, which is about prenatal testing. And as many of you know, the prenatal testing has really been revolutionized in the last few years. The vignette that goes along with this chapter, I think is quite interesting, and I'll try to give a quick summary. We were involved with the NIH and Becton Dickinson for many years looking for fetal cells in maternal blood. And the idea was that if you could get the fetal cells out of maternal blood, then with a blood sample, you could do prenatal genetic testing. And this was obviously very attractive because at the time, the testing was all invasive, either a piece of placenta or some amniotic fluid was removed with a needle or a catheter. But pretty early in the project, they dropped the idea of getting trophoblast cells, placental cells, our expertise, out of maternal blood, and went instead for nucleated fetal red blood cells that were thought to leak out of the placenta. But when they had the wrap-up meeting for the program, they invited me to come and speak. So I was really interested to go and hear where things were. I'd been following this, of course, since the beginning, and it was kind of a dreary wrap-up because nucleated fetal red blood cells. The DNA was not in good enough shape to enable any kind of regular prenatal genetic diagnosis. So it was really not a success. But at that meeting, they had also invited Dennis Lowe, an investigator from Singapore, and he gave this talk and said, look, you people, there's tons of fetal DNA in maternal blood, and I'm developing tests to use this DNA for prenatal genetic diagnosis, and this is the future. And for most people at this meeting, no one had heard this, and everyone was just stunned. Barely after he stopped talking, there people just there was barely any clapping. People were looking at each other, and the entire meeting there were little clusters of people saying, do you really think that's true? And so that was the beginning. And yes, it turned out to be true that there is a lot of cell-free DNA, and it comes from the placenta in maternal blood. So, and it's really revolutionized prenatal genetic testing, because with a blood sample now, there is a safe genetic test. So in this chapter, I talk about the difference between screening and diagnostic tests, and the fact that screening estimates risks, where diagnostic tests tell you whether or not you have a condition. And it's really important to know the difference between these, because if you get a positive screening test, this is not an absolute indication that you have a condition. It just means you need another test. So the screening tests that are around now are the non-invasive prenatal testing, NIPT, which is cell-free DNA, and then multiple marker testing, which is and can be combinations of markers such as alpha fetal protein, HCG, estriol, and inhibin. There are also integrated tests that combine markers and ultrasound. And if any of the screening tests come back positive, then this is an indication for now old-fashioned diagnostic tests, where cells of the placenta or the chorionic villi or amniotic fluid via amniocentesis is sampled and the genetic complement analyzed. I also talk about the second trimester ultrasound scan, which is an incredibly detailed anatomical scan. And most of the young people in my family and in the lab who have had this, something usually turns up looking funny, and people, it's a scary experience. You know, I think a lot of people go into the second trimester scan and very excited to see their babies, and then come out a bit bewildered when, well, maybe this doesn't look just right and we don't know what it means and it'll need to be repeated. So I think those are the kinds of things that it really helps people to prepare for. Now, the next chapter, Tracy helped me with. Tracy read this paper, made comments on this paper, and I really highlight the work of Pri in this chapter. So I hope that this will bring some positive public attention to what Pri is trying to do. and what all the members really believe in. And I'll just start out the summary by saying this was by far the hardest chapter to write. And this is for reasons that I think all of you know. It's raising the issues of the possible effects of environmental chemicals without unduly scaring people. And since writing this chapter, I have been reading more of what's in the popular press, and having struggled with writing this myself, I can see that other people who write for like the New York Times or the Washington Post also are having a really hard time framing these issues. And so as a vignette, I talk about Pat Hunt, who I've known almost since the beginning of my career, and the story of how she became involved in environmental chemical research. She was at a university in Ohio, and she studies oocyte or egg development. And all of a sudden, her control mice started having aneuploid or genetically abnormal eggs. And she just was beside herself because she couldn't do any experiments. Everybody stopped reproducing. So it took her months to figure out that the water bottles that were being used for the mice had so much BPA leaching from them that they looked milky. And it was the BPA that was causing the genetic abnormalities in the eggs of the control mice. And she moved to Washington State a few years ago, and the exact same thing happened. And this time she knew to look in the mouse cages, and this time it was a regrettable substitution. So instead of it being BPA this time, it was a substitution BPS that was in the bedding of the mice and that was causing the problems. And I'll just say, with my colleague Zena Werb, we had a grant from the California Breast Cancer Program to look at the effects of BPA and BPS on human mammary organoids. And BPS effects were way worse than BPA. So we've seen this as well. So as I said, there's a big focus on Tracy and the work of Pri, and this is the only chapter where there are lots of lists. And, you know, I didn't know what else to do, you know, stay away from this and do that, which isn't a very satisfying way to convey information, but it seemed like the most efficient. And I quote heavily from the pre-navigation guide in there and give all of you guys credit. So that's chapter 4. Chapter 5 is about pregnancy complications. And I talk about the causes and the really pitiful therapies we have when these pregnancy complications arise. I focus on preterm birth, preeclampsia, and gestational diabetes. The preterm birth rates right now in the US are about 10%, but it really depends on where you live. In the southern states and West Virginia, the rates are 12 to 13.5%. Oregon and Washington have the lowest rates at 8%. And in the countries, among the world's nations, the statistics were to me jaw-dropping. The lowest rate in the world is in Belarus, which is 4%. And this is not some statistical fluke. It's been like this for a long time. And Malawi has the highest rates at 18%. And the US is not a stellar example of preterm birth. We're sandwiched in between the Southeast Asian countries of Timor and Brunei. We're slightly higher than those countries and slightly lower than Thailand and Turkey. And right now, there's very little to be done about preterm birth. There's cerclage where you put a stitch in and physically close the cervix. But then progesterone therapy, which was formally approved by the FDA, is highly controversial. Progesterone therapy has gone up and down in popularity over the years, and it hasn't changed today. It's really, it probably depends on the type of preterm labor you have and the mode of delivery. Is it given locally or systemically? I talk also about preeclampsia. Our group has studied preeclampsia for years and years and years, and that's because the severest forms of preeclampsia are associated with abnormal placentation. This deep placentation that we talked about doesn't happen. The placenta does not get a normal supply of blood, and instead placentation is very much like in the rhesus macaque. It's just too shallow to get the amount of blood that's needed for human development. And right now, the only therapy is low-dose aspirin. So if you're at risk for preeclampsia, your clinician may suggest that you take low-dose aspirin. And after putting all kinds of money into trials of various things, nothing really works. And the only thing that's going to cure preeclampsia is removal of the baby and removal of the placenta. And unfortunately, that's still the situation. We all know that gestational diabetes, and that is diabetes that arises during pregnancy, is on the rise. Really interesting things happen around the pancreas in pregnancy. It's the only time in an adult that your pancreatic cells actually divide. So there's so many interesting questions, and we've been working on this problem with Mike German in the Diabetes Center, because if we can figure out what about the placenta makes pancreatic cells divide, it would be a new approach to therapy for diabetes. So stay tuned on that front. We're making progress. And the other thing is that substances that the placenta makes cause mild insulin resistance, where the mother's cells don't take up so much glucose. Circulating levels of glucose increase and to get more glucose to the placenta and baby. So that's chapter 5. Chapter 6 is on how life in the womb determines adult health. And this is DOAD, the developmental origins of adult diseases, a really amazing concept that's now been proven over and over again in various populations. The vignette is about David Barker and his collaborator, Clive Osmond. They realized in the early 80s, looking at statistics from the National Health Service in Great Britain, that the areas where cardiovascular disease in the north of England was rampant were the same areas where there had been a high rate of infant mortality in around 1915 to 1920. And they reasoned that if a child was struggling during that time when nutrition was poor, if a pregnant woman was having a difficult pregnancy, was not well nourished, and babies were dying because of that, they reasoned that the surviving babies were not well nourished in the womb. And because of the wonderful statistics that the NHS keeps, they were able to go and trace for many of these people, which is just, I can't imagine the challenges involved. But for the people who were developing early onset cardiovascular disease, they went back and traced their birth weights and found they were all very abnormally small, low birth weights. And this caused them to develop what's now called the thrifty hypothesis. So if a baby is not getting adequate nutrition in the womb, then the baby becomes thrifty. The baby does what it can with the little nutrition it gets. But that means, unfortunately, that once that child starts eating normally, then it's very likely they're going to gain weight and their adiposity will increase, increasing their risk of cardiovascular disease. And I won't go into it, but this theory has been proven in many populations, in Northern Europeans, in Indian population, in China. So it seems to be a property of us as humans and not just certain populations. We think that the placenta, because the placenta is feeding the baby, plays an enormous role in fetal programming. And this really brings up the specter of epigenetics, which are modifications on DNA that control whether or not DNA is turned into RNA and RNA products such as protein. Many people now think that epigenetics is modifiable by diet and metabolism. There's lots of proof for this. We have proof for this in our lab. And the epigenetics could be coupling metabolism of the mother to placental function and imprints on the baby. The final chapter is about birth, and it's really, I start out with the vignettes with the fact that we tell our kids so much about everything. Our kids know all the body parts, what the body parts do when they're very young, but very few people know about how they were born, or even how close family members and friends were born. And I think it's important to share these stories. And it's particularly important for mothers and daughters and family members, because the type of birth experience you have can be inherited. And I talk in the book about not only is the placenta why human birth is difficult, but also the mechanics of human birth. So if we look at the gorilla pelvic outlet and the gorilla head, you can see there's no problem there. That head's gonna fit right through the pelvic outlet. But in humans, it's a race. Our head is so big that it barely squeezes through, which is why many babies' heads do look squished. So this is another obstacle for easy birth in humans. And then I talk about things that I think that people might not know. For example, most women opt for serious methods of pain control during birth. And I think, you know, there's almost some sort of shame associated with this. And I go over the statistics, and hopefully it will make it more acceptable for excuse me, women to think about pain control. And I also talk about why home birth is a bad idea. And I know this is not going to be a popular concept with many people who are committed to it, but it's the statistics are very stark, and I think people should know what they are. And the stages of labor are also, I cover, because I think it's really hard to know what's going on when you are having a baby. And finally, I end by somewhat on a humorous note, by the many reasons why you should not eat your placenta. And then there are lots of acknowledgements. My friends and my husband contributed to this book. And I've already mentioned Tracy's big contribution, and my colleague Mike McMaster, my MFM close friends, Glenda Jacobson, Chris Redmond from Oxford, who's a preeclampsia expert, and Mike German here. And my husband, who loves development and loves medicine. So he was a good final editor. So thank you for your attention.